I've jumped on the nanotechnology bandwagon, since Gang invited me today, I'm going to be talking more about our work in the biotech area, I think, in nanotech area, than biomaterials and tissue engineering. I'm going to save that talk largely for Hilton Head and my golf clubs. So I want to give you two examples of how we've been trying to merge the capabilities of an area in material science that is focused on intelligent materials with applications in biomedicine. And in material science, there's actually a pretty well-developed wing of that field that's been concerned for many years with the development of a class of materials called intelligent materials. And I know when I first came to bioengineering from my background more in biochemistry and biology, I was a little skeptical about how smart these materials really were. But I've become convinced that they're pretty darn smart. And the basic property of a material that is termed intelligent is that it can sense and respond, and it can do so reversibly. And so upon an input or some sort of stimulus, an active or intelligent material then undergoes a change in its structure function properties to provide an output, a response type of signal or change in properties. And a great example is a strain sensor where the input is mechanical deformation that leads to some output and signal called voltage, and the degree of strain modulates the degree of voltage, and so you have this coupling between a signal and a strain, and these materials ideally are fully reversible, so it can sort of be a reporter of strain. Our own interest has been to couple these kinds of active materials that change their properties in response to environmental signals or changes with the control of biomolecular activity, such as protein binding, enzyme activity, DNA hybridization. And so what we're really talking about in this case is a set of materials that are polymer-based rather than a kind of bulk material. And so the biomolecular materials that I'd like to describe to you are hybrids between smart polymers and protein or DNA. I'll focus on protein today. And what we're interested in doing is coupling the changes in these smart materials, such as changes in conformation, shape, volume, their mechanics, changes in signal output, chemical properties, and use that for applications such as switching bioactivity, controlling enzyme reactions to couple to biosensor signal detection, a process that we call biomatchmaking, where in complex mixtures of biomolecules and, for example, diagnostic applications, you can control when a particular component is turned on or off, when and where two particular components such as DNA and protein come together, and to do that with a time-controlled element so that you can do it sequentially from a complex mixture. And this really isn't possible to date with current technologies. When you put molecules in there, there aren't any really good methods for controlling which ones are on, which ones are off. And so things usually have to be sort of controlled temporally in terms of which components are there. You can't just start out with everything. You have to add them sequentially to control that time axis. So in the first part of my talk, I'd like to address this development of what we call smart biomolecular components or materials that control when and where molecules come together in devices, and when and where their recognition properties are turned on or off. That's sort of the functional part of it. And the polymers, as you'll see, serve as both an antenna to to allow us to couple in. This is sort of the talking part from our viewpoint. And it's the listening element from the biomolecular material component. So this provides a listening capability that we can send signals into. But because they're active materials and change their properties, that change in properties represents an actuator kind of function. And so they both sense and then they can talk. And so they kind of listen and talk. And this change in, say, as I've just drawn the cartoon, this change in conformation from more extended, higher volume conformation to a smaller conformational change can be used to, for example, in this simple cartoon, block the association of two proteins. But in the collapsed form, then sterically, the two proteins can come together, and this represents an on state, whereas this is an off state. And we've really become particularly interested in biomims and microfluidic applications of these kind of capabilities where you could, for example, in a microfluidic stream, again inject complex mixtures of proteins and control where in a stream by controlling where this signal that converts this polymer into the collapsed state occurs. And so it may occur at a given spatial location compared to where they're injected. So a little more on these smart polymers. They exist in one state as an expanded coil that's very hydrated. And depending on their composition then, with small changes in pH, temperature, as I'll show you with specific wavelengths of light, you can drive an entropy directed collapse that's energetically driven, again, by the entropy of water release. And so this collapse state can be generally considered as a more hydrophobic state. In this hydrophobic state, they can actually also aggregate, and this is also very interesting for microfluidic applications because your diffusive properties of these aggregates change, and they can do so reversibly. So you can bring molecules together if you hook them onto these polymers or drive them back this way. Or in some applications, you're just interested in using this conformation to control activity. Another property of these smart materials, though, is how sharply they interconvert between these two states. This is one of the best studied of the smart polymers, N-Isopropylacrylamide. This is the chemical structure here, and it's this poising of water between this alkyl, more hydrophobic group, and where it can hydrogen bond to these positions that drives this sharp entropy change as a function of temperature. And so you can see over just 2 degrees Celsius, you go from a clear solution where these polymers are expanded and isolated polymers to this collapse and subsequent aggregation that then scatters light and becomes turbid. And this is fully reversible over this temperature range with N-Isopropylacrylamide, and you can see how sharp that transition is. So it's what's called a critical event. It happens, it's a very sharp switch. Now, if you copolymerize NiPAM with more hydrophilic monomers, for example, such as acrylic acid, you can shift at what temperature and control at what temperature this transition occurs, and that will also affect and sometimes broaden it out a little bit so that you can also, rather than just a pure switch, think about intermediate conformations between this and that. So our first idea, and this illustrates the concept, was to couple these smart polymers, and this is a cartoon of a protein here, near the active site of a protein. So this may be a ligand, it might be a substrate if this was an enzyme, and that we would position this responsive polymer at a special location that was fairly near the active site. And when we then triggered this reversible change to the collapse state, as we initially drew this up, we pictured that you might affect the off rate so that you could eject a ligand, or you might block the reassociation of the ligand with the protein just sterically. And what we've demonstrated is that this general concept is valid. And by controlling, again, the composition of this polymer, you can do this with changes in pH that are 1 to 2 pH units, so very friendly to biomolecules, with light or with temperature. And again, concept is that the polymer is the listening device, the antenna, and the actuator to control this active site to turn proteins on and off. In a little more detail, we've done a lot of work, initial work with a protein called Streptavidin that's one of the most used in the biotechnology field in diagnostics and laboratory assays. And this is a molecular model of Streptavidin shown here in purple. And what Streptavidin does is bind this small molecule called biotin, whose chemical structure is shown here. And from the X-ray crystal structure, you can sort of see the biotin in here. It's a tetramer, so there are 4 subunits here, each binding 1 molecule of biotin. What we found is this is a model of N-Isopropylacrylamide that's approximate molecular weight that we've been working with. And this is sort of a schematic to show you that if you hook it up here near the binding site, you can sort of picture how it can serve as a gate to the association of a molecule into this active site. And so the polymer we found can serve as a very effective gate to prevent the association of biotin onto the protein. What we've also found, on the other hand, is that we're not very good at, once biotin is bound, at ejecting the biotin, so getting biotin back out. And if you really look at the structure, you can sort of see the difficulty here. This is a very buried binding pocket, and if I really showed you all the atoms on the protein, you wouldn't see biotin at all. You'd just see this very end, this carboxylate group, which is out on the surface. And so the polymer can't really access this more buried binding pocket, and so it's been very difficult for us to come up with a way of coupling this change of the polymer to the ejection of biotin, because this end isn't really attached, and so you don't really exert any force on this kind of scheme. Where we have been able to manipulate the binding in this buried pocket is by using a polymer that is not only just end reactive, but has reactive chemistries, chemical moieties scattered along the backbone. And in this case, we engineered a unique cysteine residue at this position shown here relative to the binding pocket, and it is directly adjacent to one of the most important binding contacts, a tryptophan at position 120, that we knew from other studies was very important in this binding of biotin, and so we positioned this attachment site here. But because this polymer can be grafted at multiple points, what can happen is, this is just schematically, the polymer can be attached at multiple points such that when it collapses, it can actually generate force. And what we've seen is that this is the only way, by using this pendant polymer, where we are cross-linking at multiple points, that we can get release. And so this is a control where we just look at biotin, the amount of biotin bound on the y-axis as a function of temperature, so you're going between the expanded and collapsed state of this polymer. And the control just tells you that there is no temperature dependence of biotin binding, and that's the problem. There's no reversibility to this binding. On the other hand, with this pendant polymer, we can at least begin to see some release. When we collapse the polymer, we lose about 50% of the bound biotin, and this can be cycled so that eventually, if you go through many cycles, you can get rid of all the bound biotin. And so it shows you the capability that you have in controlling how the polymer is attached. We control where it's attached. That's another important design parameter, and sort of generates this model that got us going. Okay, what I want to give you are some unpublished studies where we've looked at a sort of a different class that I think is much more amenable to this idea of proteins that have exposed pockets. And so the problem here is that the polymer doesn't have any access to this buried binding site. But if we look at an enzyme that I want to talk in more detail now about, an enzyme, here many enzymes and many other types of proteins, such as antibodies, have a much more exposed pocket. And these are 3 residues in this particular enzyme that are the important catalytic site residues. And this entire groove, it defines a large open groove on the exterior of the enzyme that's accessible to polymer. And this is a very different setting, and we've, to anticipate the results, saw a lot better switching activity. So in this particular example, what we're after is generating our first photoresponsive switch and our first application with enzymes. The idea, again, is similar. Now this becomes an enzyme, but it becomes a bigger, more open cleft for the substrate to come in, but it has the same idea. With the photoresponsive polymers, what we're after is switching in different directions with different wavelengths of light. So for example, you could go to this off-state collapsive polymer with far UV irradiation at 360 nanometers, and then go back the other way with a distinct wavelength of light of, say, 420 nanometers. And after a lot of work of a very talented graduate student, he developed such a polymer. And this is the chemical composition. It's a copolymer of dimethylacrylamide and an azobenzene moiety shown down here. And he actually developed, and I'll sort of be giving results, of 2 different classes within this general family of copolymers where the azobenzene group is attached either through an ester linkage, and I'll call that the DMAA, I have to really remember that, or an amide linkage, DMAAM. And the important thing about this, I'll show you in a second, is that for reasons that we still don't understand, that subtle change in ester versus amide linkage give these 2 polymers opposite photoresponses, so that you can either go in this direction with UV and back the other direction with 420 nanometer light, or vice versa. You can go in this direction with visible light and go back the other way with far UV. This is driven just really quickly by the trans-cis photoisomerization of the azobenzene moiety, and why we think that this is coupled to this more macroscopic change in the polymer going from expanded to collapsed is the fact that in the trans state there really isn't a very significant dipole moment which affects water ordering again, whereas in the cis state you have a much larger dipole moment, and that's coupled again to driving this water release that underlies the change in the polymer conformation, and it's fully reversible by these different wavelengths of light. So this is what the sort of temperature and photo responses of these polymers are. It's a little complex because they're both temperature and photo responsive, which is very useful from an engineering standpoint. So again, we're looking at turbidity of solutions of these polymers so that when it's clear, you're in this expanded, hydrated, isolated state, but up here you're in the collapsed state where they aggregate and scatter light. And so you can see, first of all, that we've selected the copolymer composition so that this transition occurs at around 40 degrees Celsius. And that was chosen very carefully because that's where this particular enzyme target has its maximal activity. But the interesting thing is that if you study where this transition occurs, under UV versus visible light, you can see that with this particular polymer, this is the ester linkage, the UV light increases the LCST. And so down here, somewhere in this range of slightly above 40 degrees, if you look vertically at a single temperature, you have this maximal difference where the polymer is more collapsed up here and less collapsed here under UV versus visible light. And so at that isothermal temperature, by shifting the polymer between these 2 photoradiation states, the polymer exists either more collapsed or expanded. And the interesting thing that we still don't understand very well is that the amide-linked polymer goes in just the opposite direction. So UV lowers the LCST. So you can just go in the opposite direction, which may be important for some applications, but not so important theoretically. So we've been working with a model enzyme called endoglucanase 12A in collaboration with Genencor International, which is a big producer of enzymes. And you maybe I certainly didn't appreciate what drives Gin and Core's business. They're the second largest enzyme. Does anybody know what drives their business? Bulk enzymes? A couple of things. One is laundry detergent. If you've seen the commercials where it has some special component that degrades protein stains, food stains, they put enzymes in there, and it's mainly a marketing thing. The other big use for endoglucanase enzymes, what these do is degrade cellulose. And so what these are used for is in making your jeans look like somebody else wore them before you, by degrading to a fine degree of control how much fraying, how much sort of stone washing, you know, what kind of look that they have. And so they enzymatically degrade the cellulose fibers, and that's what these enzymes do. And these are the catalytic site residues that are involved. And this is just a model to show you roughly kind of what size we would expect for these stimuli-responsive polymers relative to this particular enzyme. And what we do is go in and genetically engineer handles here by putting in cysteine residues at defined positions and then hooking the polymer up. And by deciding where you put this hook, you can move the polymer sort of closer to the enzyme active site or farther away. And this is a bigger, better cartoon. This would be a model of the substrate. It's a really cool enzyme. It kind of looks like a hand, and the hand just grips around the cotton fibers, and that allows it to degrade along the fiber length. And so you can see relative to the substrate, there's a very exposed pocket. The idea is that in the expanded state, the polymer would not interfere with that. This would be the on state, but with, say, a change in temperature, or I'll show you photoradiation, you would collapse the polymer, and it would sort of block off this substrate from being able to get in there, and that would be the off state. We found that there are two key parameters, the conjugation site and the polymer molecular weight, in terms of the molecular bioengineering that's part of this. This shows you a little better side view so that you can see that the fiber would be pointing into the plane of the screen here. And it's kind of a hand that wraps around that. And then I'm going to show you some results by conjugating the polymer at this position, which is very near and located sort of down around this axis, so that we picked this position when I designed these so that the fiber couldn't get into that, fit all the way through that groove so well, whereas this one position was moved a little bit away. And then we also have a control where we've conjugated to this N-terminal position, which is the farthest away from the binding site. We use pretty standard chemistry. These polymers have been end-modified with a sulfhydryl reactive group, the vinyl sulfone group, so that the cysteine that we engineer into the enzyme can react then just with the end of the polymer. So this is an example where the polymer is just engrafted onto the enzyme. There are two application formats and two that are commonly used in all these devices. In some of these, you want to use an immobilized conjugate, and there's a number of different ways that we hook these polymer protein conjugates onto, say, magnetic beads. The chemistry isn't so important. But we can also then view this as a switch for free proteins that might be in a microfluidic device, for example, or just in a laboratory assay, such as PCR or something like that, where the conjugates are brought together into aggregates. And this aggregate, what we've seen, for example, is in the off state. But again, it's reversible to send it back out to this state where the polymer is more expanded, and this would correspond to the on state, just free in solution. Well, how well do these work? We were surprised because we worked so hard with Streptavidin for so many years, and I really think it's a function of this exposure of the binding site, but these work really as spectacular switches. So this is with a small molecule substrate called ONPC at 45 degrees Celsius, like I showed you at this sort of optimal isothermal condition. Sorry, this is temperature. This should not be at isothermal. I'll show you that in a second. This is the temperature response. At 32 degrees where the polymer is expanded, you get about 80% of the activity. This is a real drop. I mean, just having the polymer around there does knock down the activity of about 20%, but that isn't so important really. What's really important is that when you collapse the polymer, you get a nearly complete turnoff of the activity of the enzyme. And this is that position, this is jargon by us protein engineers showing that we've changed the naturally occurring asparagine at amino acid position 55 to cysteine. And so this is attached very near to the active site, and that's what is going on there. This S25C is moved a little bit away from the active site, but still pretty close. And you can see the general trend that as you move it away in the off state here when you're collapsed, there is significantly higher activity, about 20% activity. And then when we move it predominantly to the N-terminus, this is an ideal control. What we'd really are trying to do is make a control where we're attached back here. But you can see the general trend. When we move it a little further away yet, then you begin to get significant activity at the higher temperature where the polymer is collapsed. And so the conjugation position sort of makes sense just from a simple steric model. The molecular weight also shows a trend, although it didn't turn out to be so important. These are with, this is with the ester polymer. This is with the amide polymer is a jargon here. But again, with polymers of 10,000 molecular weight or greater, as we've seen, you get this nearly complete turnoff when the polymer is collapsed. As we go down to a 6,000 molecular weight polymer, you begin to see more activity in that off state, and a 3,000 a little higher yet. And when you go all the way to just physical mixtures, you see no difference in activity between 32 and 52 degrees. And so it does require conjugation, and there is a molecular weight effect, although even the 3,000 molecular weight polymer at these positions works pretty well. What we're really interested, though, is the photoresponsive, and so I spliced this in incorrectly. Now, this is the isothermal condition, so we're at 45 degrees, where there's maximal difference in the polymer being expanded versus collapsed under visible versus the UV photoirradiation. And so with the ester linkage, I'll just remind you, under UV conditions, the polymer was expanded, and what we indeed see is that that's in the active state. Now, what's interesting is that here we have lost about 40% of the activity, which still actually isn't, from an application standpoint, really all that significant. It's still active enough. But what's important is that when you collapse under visible photoirradiation, go to this state, it's a pretty darn good switch. And the same with the amide link. You go between the on state and the off state, but in this case, the off state is UV versus visible. And this is a control, interesting control, where we just put that azobenzene monomer, so it's not the polymer, just the small molecule, onto the same position. And you can see that the small molecule photoisomerization does not act as a switch. So it has to be a larger polymer to get this kind of switching activity. And in terms of the reversibility then and the cyclability, these are also really beautiful and much better than anything we ever saw with Streptavidin. And so if we look at Thermo Cycling first, what we're plotting here is total product accumulation as a function of time. And so you're just sitting there with a cuvette with your conjugate in there, and you switch it between 32 degrees and 52 degrees, and this is kind of like real time here on the axis, and you just switch it back and forth and measure product accumulation. And so both of these polymers have the same temperature response, both the amide and the ester, so you'd expect them to be the same. So at 32 degrees, where the polymer is expanded, you accumulate product. The enzyme is sitting there working away. But when you shift it to 52 degrees, just in the time that it takes, this, the fastest we've looked at is sort of seconds here of manually shifting this to a 52-degree water bath, it's turned off, and it's turned off indefinitely for as long as we've looked at it. But you shift it back to 32 degrees, and now your switch is back to the on state, and so you can accumulate product and you can turn it off. And you can keep cycling it like this, so it's a real-time switch. And with the photocycling, we've seen similar properties, but the two polymers now have opposite responses. So under visible, this particular DMAA-M polymer is in the on state, the expanded state. Shift it to UV, again in real time, it's off, on, and off. And the other one just has the opposite photo response. So these conjugates really show the properties of what we were after initially in terms of being able to turn things on and off in reversible fashion. So in summary, where we're really heading with this are in applications such as microfluidic streams, and it holds not only for proteins, but we think also have some very early results with DNA that are interesting. But again, you can control sort of which components come together, such as the green versus the purple. And this represents a time axis, because at this point, if you're shining light at this position of microfluidic stream, then the purples may be in the on state and working, whereas the greens are turned off. And this is totally reversible so that you can capture DNA molecules, release them at different time points in a more complex device environment, or maybe just a laboratory assay system where there isn't necessarily have to be a microfluidic stream. You can turn enzymes on and off or just activate them. And so it's sort of like a platform capability that we see lots of applications for. It's an experiment that we're trying, and so we're initially trying it by microinjection because there are many enzymes, for example kinases, where you could have it turned off, but then turn the kinase on at a defined time with a light stimulus, for example, and that could function for a given set of time where you might turn on a signaling pathway, for example, or turn off the pathway. But we're far, I guess, from having a result. We just sort of tried some initial experiments. The conjugate in that case. So it's one of these with an enzyme where the polymer would be positioned more up here around the active site, where it would be in an off state at 37 degrees, because that's another reason why we were interested in this 40-degree range. But then with light, you could switch it on or off. It can go in either direction. All right, a second example that's a little less biotech-y and maybe a little more drug delivery is a different set of polymers. And here what we're after is a very different goal, but it shares this property where the polymer has reversible and environmentally specific changes in properties. In this particular application, what we're after is the more effective intracellular delivery of biotherapeutics. And so these are sort of the new biotech drugs that are based on DNA. It might be plasmid DNA for gene therapy. And what we're interested in is trying to make non-viral even remotely competitive with viral delivery efficiencies. Or one that we've developed quite a bit is with antisense oligonucleotides, where we're talking about small linear pieces of DNA. Or protein drugs that function inside the cell. To date, all of the protein drugs that are out there function on the extracellular face of the cells. None of them really work inside the cell directly. But there's a whole class of proteins that people are interested in and have shown by microinjection have interesting pharmaceutical properties, such as transcription factors or neutralizing antibodies, that if you could effectively get them across this barrier, you could have wholly new classes of protein drugs, and also RNA therapeutics. And the core problem is really simple. These macromolecules are not transported across the cell membrane effectively, and when they are taken up, they're taken up into a vesicular trafficking pathway where they're taken up into the endosome. And the real problem isn't the endosome per se, but the fact that this is rapidly trafficked to the degradation compartment, the lysosome, and these are all degraded in that compartment. So our technology that we've been trying to develop gets things out of the endosome into the cytoplasm, where they can then diffuse into the nucleus or reach their target from the cytoplasm. so when we started thinking about this, what we were after is a synthetic delivery system that could address this need. And it's informative to consider how biology has solved this same challenge. Viruses have solved the same challenge because they have to get all their DNA efficiently inside the cell and to the nucleus, not just into the vesicular trafficking pathway. Pathogenic organisms such as diphtheria have the same problem. In that case, it's a protein drug equivalent that they have to deliver to the cytoplasm. And they share some important properties. You can think of these as really like little nanoparticles, drug delivery systems. There's proteins on the outside of these viruses, a retrovirus example, that direct the initial uptake, targeted uptake, and so that's one part of their drug delivery system. That's pretty well appreciated, I think. What's less well appreciated is that they have a very separate component, but a very efficient component that solves this problem of getting out of the endosome, and it's coupled to a unique gradient that's generated in the endosome. They have an ATP-dependent proton pump that lowers the pH all the way down to 5 in late endosomes. So this is a large chemical gradient, and this is what these organisms have evolved to take advantage of. Out here, the proteins that destabilize membranes, biological membranes, are inactive because you're at 7.4. As the pH drops, they become activated and destabilize the membrane so that you get enhanced transport of the virus out of the endosome. And from there, then, another series of miracles happens where it diffuses or is actively transported into the nucleus, where you can then get final gene expression. If you look at this at a little higher degree of resolution, there's a fair amount known. This delivery system has a component called hemagglutinin that represents this smart component that can sense the pH. At pH 7.4, the structure of this protein has been pretty well characterized. Here it is. It's inactive. And so as we're looking inside out, this is inside the endosome. And at pH 7.4, initially it's inactive, and it's inactive as this stealthily goes through your body. But as it starts to sense this drop in pH, it undergoes a big conformation change in the exposure of side chains that can then destabilize the endosomal membrane, and that enhances the transport of the cargo, the DNA or RNA, across that membrane. And the way this works a little more specifically is that there are amino acid side chains that are protonated. They're carboxylate groups, and so their pKa matches biology. And as they're protonated, that simple change, going from a COO minus to COOH, triggers this change. And it's been well characterized. And it's well characterized also in diphtheria toxin. There's a key carboxylate residue that is the pH sensor that triggers the same membrane destabilization. So we got interested in this because we were interested in whether you could sort of learn from how these viruses were doing this, but designing it into synthetic polymers. And polymers that have an intelligence in that they can sense and change their properties reversibly in regard to pH. And the advantage of polymers is that they're not viral proteins, which are extremely immunogenic. They're also extremely expensive. And while they've shown promise in the laboratory, they're not practical from a real-life engineering standpoint. And polymers have a lot of advantages and are much more realistic to think about than these viral proteins. So I want to just close by showing you one example of a new family of polymers that sort of are mimicking this viral system. And the key is that it has different parts. As we've designed it, the backbone, shown here in orange, is going to have this capability of at low pHs being membrane destabilizing, but at pH 7.4 being inactive. And then we've grafted onto that membrane disruptive element through a pH degradable linker the actual cargo and targeting element. So a targeting ligand or protein or oligonucleotide can be attached through really simple chemistries, and a PEG group through this pH degradable linkage to such a backbone. And the key here is that we're going to generate this pH degradable, this pH sensitive element that decouples the cargo and targeting ligands from this membrane disruptive element. And the concept, then, thinking of the application, is that this is a polymeric delivery system that can target receptor-mediated endocytosis. It doesn't have to have targeting, but it can. But the key new thing is that this is a functional polymer. It's not just a carrier. And so at low pH, this falls apart, and this backbone that has this pH-dependent membrane disruptive activity can partition and destabilize this membrane and enhance the transport of these elements that then sort of break away, like the lunar lander module, and have enhanced transport to the cytosol. So these are functional polymers that direct endosomal escape of biomolecular therapeutics. Here's a first generation polymer, just to show you what the chemical composition is. And this was sort of based on some preliminary work that we had done. It didn't come out of the blue. But this is a TER copolymer of butyl methacrylate, and this is the membrane disruptive element. And this is a key part of it here. This is a styrene benzaldehyde that has an acetal linkage here. And these are the pH-sensitive chemistries. And the nice thing is that by tuning the specifics of the styrene benzaldehyde, for example, putting oxygen versus nitrogen, it affects the electron withdrawing capabilities. And you can tune these kinetics from minutes to days, the hydrolysis rates as a function of pH. So when you lower the pH, these bonds are cleaved, and these PEG groups and whatever go with them are liberated. And so this is sort of the separation, and then this is the membrane disruptive element. The design, again, was really crucial here to couple it to the biology. And so the normal trafficking kinetics and going from the endosome to the lysosome fall on the time scale of, say, 0 to 60 minutes. Okay? And so this is a time frame that you have to beat. Okay? And this is really a big challenge because at 7.4, pH 7.4, it needs to be stable. But then within 30 to 60 minutes, as this is acidifying, that has a time aspect to it, then it needs to fall apart. Okay, so it has to fall apart fairly rapidly and yet be stable. And again, lots of great work by graduate student Noreen Murty took care of this. And so the hydrolysis, if you look at hydrolysis, sort of the breaking apart, the pH dependency, it's a very slow background at pH 7.4 as the individual groups hydrolyze. So these are stable on the time frame of a day to 2 days at 7.4 before they become membrane active. But very quickly on the right time frame of 0 to 60 minutes at pH 5.4, you see this rapid hydrolysis. And that, it turns out, is just directly a function of the proton concentration. Well, how about the membrane disruptive properties? Well, we use an inside-out assay that allows you to calibrate versus other agents that have been, peptides and proteins, for example, that have been developed, where you add the polymer and you ask as a function of pH, how well does it liberate hemoglobin from inside the red blood cell. So it's kind of a model membrane assay to see how active are these polymers. Well, after a 20-minute incubation, and this is really, again, out to days, at pH 7.4, you don't see enough hydrolysis to give you membrane disruption. But at pH 5, after just 20 minutes, this thing hemolyzes all the red blood cells under these standard conditions that are sort of a gold standard in the field. And so what this means is that this polymer will liberate all the hemoglobin from 10 to the 8th red blood cells in 1 mil in PBS. How does that rank? It's actually 3 to 4 orders of magnitude better than the best viral protein. And we've seen this with a whole set of these polymers, is that they're actually more active at membrane disruption than the biological proteins are. It's kind of surprising, but you have this amplification effect because they're large polymers versus much smaller proteins. just to give you one example to show you that these really sort of do work, and we've done plasmid DNA, we've published that, and we've also published just recently, I think it's just coming out with a protein, but I wanted to show you a biomaterials application. One of our big interests, and I'm sure you heard about this last week with Buddy, is a control of the macrophage phenotype and control of activation. And one class of drugs that we've become very interested in are antisense oligonucleotides that knock out specific gene product or the messenger RNA that are tied to particular gene products. And so what we've done is just modify the PEG groups here with mannose that targets mannose receptors on the macrophage. And in this case, we've just put on lysine groups to electrostatically complex the negatively charged antisense. So this is the full carrier. This is a group that is then designed to be, once the macrophage takes up these polymers, to hydrolyze and liberate the cargo, and this would then enhance the transport out. And it turns out that the macrophage has been a really tough model, and that was the other reason why we wanted to try this, because while macrophages are very efficient at taking things up, they have an unusually high concentration of degradation enzymes. So it's been very difficult to get antisense into macrophages. It really hasn't been very possible practically to do that. And also the same is sort of true with proteins and DNA. And our particular target, it was done in collaboration with Ron Mayer, who's the a surgeon at one of the big hospitals in Seattle at Harborview. And one of his residents, Joe Kucheri, and they identified interleukin-1 receptor-associated kinase as a good target for antisense therapy. It plays a key role, and this, believe me, is an abbreviated signaling pathway representation, but the key thing that's tied to downstream that we're going to assay for is tumor necrosis factor, and we wanted to knock this down by knocking out this IRAC transcript. And IRAC is a good candidate because while it's tied to a phosphorylation event that regulates its activity, it is ubiquinated, and so it's degraded in the proteasome. And so if you knock out the transcript, you can knock down the levels, unlike a lot of other intracellular kinases who are sort of constitutively produced and present. So there's a lot of data here, but I mainly want to point out that what we're looking at for each of these samples is what a non-LPS stimulated versus LPS stimulated production of, sorry, this should be TNF here on the y-axis. So when you stimulate, normally you get a big upregulation of TNF, and we want to ask, can this anti-IRAC oligonucleotide knock that back down? And the polymer, and we've seen this in a lot of different toxicity assays, and it's also been through some very initial in vivo screens, has very low toxicity. And the reason is because at 7.4 it's not active. Okay? And so this was a good sign. The polymer did not sort of super stimulate further TNF production, which these cells are very sensitive to. Another control is the sense strand. So this is not an antisense, and it can hybridize and lead to degradation of the target mRNA, so it's up there. The antisense alone may have a slight effect, but this is the problem. When you just put in antisense oligos, they're not efficiently delivered, and so you don't see really any knockdown in TNF production. But finally, if you look over here, with the antisense plus polymer delivery system, we get very significant knockdown of TNF production. And so we've seen this with several different types of antisense, also in the macrophage and other types of cells. And so what we can say at this point is that in cell culture, these seem to have unusually high activity in getting antisense. And we've also, in different cell types, like I said, done proteins and DNA. The concept is sort of general for all these classes. So in conclusion then, we've developed, I didn't have time to really show you, but there are really a whole different families of chemistries of these polymers that have the same property of coupling high membrane destabilizing activity with controllable pH profiles. And they're very interesting because you can control, does this happen at pH 6.5 or does it happen at 5.5? And that's important biologically because this is a really gross oversimplification. The endosome is evolving at all times toward the lysosome, and they're different biologically. And it may be better to disrupt early, or it may be better to disrupt later for all we know. And so you can investigate that by controlling at what pH these become active. And we've seen that it enhances gene and protein delivery with low toxicity. This was a polymer I didn't have time to talk about, but the encrypted polymers say the same. So just in conclusion then, I would like to acknowledge certainly my good colleague Alan Hoffman. We have a joint group working in these areas. Noreen Murty was a person who did all really of the work that I showed you the second. I'm giving his name to Bob because I think he's out getting ready to look out for some academic positions. Zhongli Ding did, and Tsuyoshi Shimaboji did the smart polymer protein work that I discussed. And we have many nice collaborators, Ron Mayer and Joe Kusheri in particular on that antisense example were very important to us. And this work was funded primarily by the NIH and also by UWeb-ERC, where we're interested, show here, of delivering these kind of drugs from scaffolds and biomaterial coatings so that you can enable a much broader class of potential therapeutics to modulate, say, macrophage activation. So that's it. Thanks very much.
We got started a little late. I know some people have class, but let's take some questions. Okay.
So really very exciting. For delivery, could we just bypass the endosome altogether?
Well, that's sort of the competing technology in a sense, I think. Or that's what's always asked of me is, how does this compare to the TAT peptides and these peptides that apparently have some ability to move macromolecules directly across to the cytoplasm. And those are exciting, and you know, the way I look at it is that we can easily use those on our polymers, number one, because we have a modular system where we can put targeting ligands or a TAP peptide onto our polymer. The problem with those peptides, on the other hand, is that they're nonspecific. And so for in vivo use, you really have to worry about those peptides because the first cell that they get to, they will, if they're working as advertised, which I have some questions about, they will go across that membrane. And so in vivo, you have to have some way of sort of introducing specificity to your delivery system. Now in vitro, they seem very attractive and probably very useful. And so, but for in vivo, you have to worry about that.
So if we add a target ligand to recognize specific cell type of disease cells, combining that with the peptide.
Well, yeah, I think that's an interesting question, gang. I think the question is which dominates, though. And so even though you have a targeting ligand, it will depend on the kinetics of the bioavailability, biodistribution as to how quickly do you partition to the target site versus along the way, that thing is bouncing into other cells, and if the TAP peptide is there, then there's no reason why it couldn't transport still, even if it has a targeting ligand for a different cell type. So, but all that's really interesting. I think it's so emerging right now that it'll be interesting to see how it plays out.
Great.
Really outstanding work. Many of these gene delivery systems work very well in vitro. Then there are issues when you try and do it in vivo, clearance and inflammation. Viral vectors are a clear example. In vitro, we can get 99% infection efficiency. Sometimes when you don't go in vivo, it just gets clear. How do these polymer backbones stack up in terms of clearance?
Well, we've been in the in vivo studies that we've just started. are in two areas. One is with plasmid DNA, so it's a true non-viral delivery system. Now, in this case, the polymers are packaged together with a non-viral system that has a condensing agent and the plasmid. And so the polymer that we have is an additive that's sort of, again, equivalent to hemagglutinin. Well, we hope it's the way the formulation turns out to be very important, and we actually think that it's important that you formulate it in last so that it's available and coating the surface. But what we've seen is that it does not significantly affect biodistributions because it's just mainly it's the condensing agent in the DNA. And so this is an interesting case where it may actually not have such great in vitro properties, because in vitro you don't have to worry about biodistribution. What people have found is that you cannot use these lipids that look great in vitro in vivo because they get taken up by the RES system, for example, and rapidly cleared. To get around that, you pegylate. But then the pegylation really kills you on uptake at your target. And there again, then this escape becomes a very important parameter. And what we've seen is that the polymer, in these cases where the system has been optimized for biodistribution but exhibits poor uptake, the polymer then becomes very important in vivo, okay, in the one study that we've kind of been doing. And so it's a very complex from an engineering standpoint, because all these different parameters play off of each other. And it's still yet to be seen how well our polymer really enhances, but it's for sure needed in the non-viral because it's a real barrier getting inside to the cytoplasm. Now, in the antisense case, it's kind of interesting because there, when you attach it to a polymer, has completely different biodistribution than the antisense alone. And this can be either an advantage or a disadvantage, depending on your application, but you got to play to your strengths. And for example, polymeric drugs accumulate at tumors because of the leaky vasculature. And so you just have to take that into account. And they have completely different biodistributions in the antisense alone. And you need to, that will kill you for some applications. It will open up possibilities in other applications.
Serena?
Well, see, I think that's where from a design standpoint, we've always said it has to be reversible, because if you have much membrane destabilizing activity, and these have potent destabilizing activity at pH 7.4, you're in trouble, you know. And so the key thing about these polymers is that as they reequilibrate back, say in the cytoplasm, once the endosome is disrupted, or it's not, we don't even know if it's really disrupted. It's destabilized is why I choose that term carefully. That at pH 7.4, they're back to being very peg-like. They're hydrophilic, and we haven't seen any toxicity. We haven't even been able to get a toxicity profile, because at 7.4, there's no toxicity. And I think that's the key thing, that inside the endosome, but only at that low pH, are they active. Now, there are additional, more complex questions as you accumulate these and you destabilize endosomes too much. Then there's endosomal storage diseases where that may evolve over time if these things are accumulated. And so I think the bigger question at this point is clearance. How are these cleared out so they don't accumulate inside of the cells? And that we have not gotten into yet. And of course, it's a new Ph.D. thesis topic to make these biodegradable, right? But still maintain these properties. Can you do that? Well, it's fun academically to think about. But I think that my own feeling is that they're not going to be too toxic, and there are many applications where you don't have to worry about just continuous big delivery problems, that you can go in with a relatively safe amount, and they will get cleared just like PEG does. PEG's approved.
One last question. True in.
Because you can have many of these fungi modules per polymer backbone, so have you observed some kind of effect like multi-valency effect or chelation effect? And you tested that, it became the range these kind of fungi modules very different.
Now, is this for the first part? You mean with the
For the second part.
for the second part? Well, they're random copolymers, and so they do carry multiple doses of whatever the antisense. Is that am I maybe I'm not understanding your question correctly?
Membrane disrupting elements.
Uh huh.
For example, peptides. Let's say you have 10 peptides all put on the same polymer backbone. Would that be equal to one peptide for each backbone?
No, it's better. It's cooperative, and it
That's the multivalency.
yeah, absolutely.
How big is that?
Well, it's a huge effect if you want to go in that direction. Now, we've tried to optimize our polymer so it's the disruptive element. But it's kind of interesting. You can couple it with membrane disruptive peptides that are based on these viral proteins and see that at concentrations where those peptides have no activity whatsoever, that if you conjugate an equivalent concentration of the peptides, they're completely membrane disruptive. And that's because you're saving the system entropy because they're already joined together, and there's no entropy loss in associating those peptides in such a way when they go into the membrane that they're disruptive. And so it's very much a cooperativity effect in that case.
So that's the trapping effect, right? If you only have one leg in a crab, probably won't trap anything.
Well,
Two legs won't trap anything.
yeah, there's an analogy, but I don't think it's multivalency. In this case, it's not actually multivalency. It's because what these peptides have to do is assemble into a membrane disruptive assembly. And in that case, it's a question of, is the assembly favored? What concentration do you have to drive that assembly of those peptides to associate and aggregate together to become disruptive? If they're isolated, there's an entropy cost to associating them. But when the polymer is brought into play, then it's intramolecular assembly. And so it's not multivalency, but it's more of this entropy cooperativity effect rather than multivalency. But I understand your analogy to multivalency.
Two of you are going to lunch together. You can talk about crabbing at lunch. I think we need to bring the formal seminar to a halt. Pat, thanks very much. It's
Thanks, Bob.
a great
It's
seminar.
always fun to be
Thanks,
here.
everyone.